So let's wrap up talking about sensory restoration, but now we're going to turn our attention to sensory restoration in the domain of vision. So we talked a little bit on the methods day when we talked about optogenetics about how sometimes people will lose their vision because of this disorder called retinitis pigmentosa. If this is what a normal retina looks like, this is what a degenerative one looks like. Over here, you can see these nice rods and cone cells. Over here, you can see it's kind of a jumbled mess. So a question that people have asked in terms of visual restoration with brain-computer interfaces is there a way that you can interface directly with this neural circuitry? And the idea here is similar to the idea that people had when they were doing optogenetics in the water maze that I was showing you, which was, okay, well, sure, the rods and the cone cells might, not be functioning anymore because of the degeneration, but the ganglion cells underneath that lead to the brain, they're still there. So maybe what we can do is interface with the remaining functional circuits that are on the retina and sort of replace the rods and the cones with this brain-computer interface. And so in this case, it's actually it's very similar to what we were talking about before. You have these little, very tiny electrode arrays. And to kind of give you a sense of really how simple the technology is, or at least the process of it is I think maybe hopefully some of you when you were kids, you play with these toys. I don't know how to refer to them, but they're these needle-ish toys. I used to think they were so cool when I was little. And you can press them in different ways and you make different shapes. It's the exact same type of principle where you put an array of electrodes like this to make different shapes right on the retina. And so actually here are a couple of different images of people who have had, and these were really prototypes, but they've gotten much more sophisticated since then, directly implanted on the retina. So the way this system works is that you would have someone wear, glasses in this case that are connected to a camera. The camera is connected to the microelectrode array that is right on the retina. And then this microelectrode array, it can't activate the rods and the cones because you can see some deterioration down there. But what they will do is they'll activate the ganglion cells and all the other cells that are on the retina that themselves do not respond to light. But you can therefore have these little electrodes be the responder to light. This to give you a sense of what it looks like when it's fully functional. Here's an X-ray and from a person who has this in their eye, and literally it's just, again, you implant this directly on the back of the circuit. Okay, here is just a little video just to drive this home again and make sure we're on the same page before we move forward.
In Second Sight's Argus 2 retinal prosthesis system, a miniature video camera in the eyeglasses captures the scene. The video is processed by a small portable unit and transformed into instructions which are sent back to the glasses. These instructions are then transmitted wirelessly to the implant on the eye. The implant consists of a receiver and an array of electrodes. Instructions are received and corresponding signals sent to the array, which emits small pulses of electricity. These pulses stimulate the retina's remaining cells and are transmitted down the optic nerve, conveying visual information to the brain, which perceives patterns of light. Patients learn to interpret these visual patterns.
So the way that this works is just therefore really basic points of light, okay? So for example, if you are a patient looking at a person like this, the way it will appear to you is like so. And this is pretty crude, this is pretty elementary, but if you are a patient that was completely blind, this is a big step up. You can identify big objects at high contrast. You can recognize, doorways and when someone's moving or approaching you or so on and so forth. But of course, you can't recognize one person from another person. You might struggle with identifying things and any kind of clutter. And actually, I wish I could find a video of what this looks like more dynamically, because as you move around, if you move around and show multiple samples of this, the fact that it's a dynamic video also gives you some cues of where things are. Now, the video that I showed you all a second ago had, what was it, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 by 1, 2, 3, 4, 5. It was like a 60 electrode array. Over time, though, these have gotten way more complicated. And the more complicated these get, the more electrodes you're able to get, the more precision you get. So for example, if you just have a 4 by 4 electrode array, you can't tell a whole lot. But if you get, 100 by 100 electroarray, now you can actually start to not only tell that this is a human, but you can tell that it's a bald man, that he's wearing glasses, that he's facing down to the right, and so on and so forth. So let's show a video of what that looks like in action.
To try to imagine how it might look to Alan, Dr. Iezzi says to picture contrasting light and dark blocks on a grid.
But by moving his head and using his visual memory and all of his cognitive skills and his remarkable capacity to get around, Mr. Zared can reconstruct a scene.
He just says reconstruct a scene, sorry. So this is what I was saying about if I could have seen a video where it's kind of dynamic and moving around, you can tell that like this static image here isn't as like interpretable, but when you put dynamic elements to it, it becomes much more interpretable. Okay, now sometimes though, people will not be able to have their vision restored because of the retinal approach, okay? Sometimes the damage is not just to the rods or the cones. Sometimes the damage could be to the eye itself, the retina itself, the optic nerve, and so on and so forth. And so in that situation where you can't just innervate with something like the ganglion cells, what people will do is they'll take a cortical approach to restoring vision, okay? And in this case, what they'll do is they'll put the microelectrode in a part of the brain, for example, like V1. And so what you can get are situations like this where people have these kind of permanent electrode arrays implanted into the back of their head. And so in a situation like this, the camera will process information that's out in the world, will send a signal to the electrode that is in V1, and then V1 will get stimulated as a function of what is being processed by the camera. I want you to remember now that we talked about how early visual cortex in V1 has a thing called retinotopy, where we talked about how, you know, when we talked about that situation where the monkey looked at the image, I wish I put a slide in here, sorry I didn't, the image of like that checkerboard spiderweb grid, it would show up right on their V1. Neurosurgeons are able to take advantage of this because since there's this map on your visual system that corresponds to the world, this retinotopic map, what you can do is you can activate something like a tree pretty easily by just activating that retinotopic map, okay? So this is basically the approach that people take where they're trying to restore vision with BCI or BMI, but there's also ways in which people have tried to restore vision with gene therapy. One case is obviously optogenetics, which I've already referenced. In that situation, this is like what we talked about from way earlier in the course, where rather than try to innervate with these cells that still are on the retina through BCI, you could do it with optogenetics. You can't really do this with humans right now. Right now, humans are, not cleared to get optogenetics done in them because there's long-term concerns about will they predispose, for example, to the development of cancer. That's one of the reasons why brain-machine interfaces or BCIs have gotten created. But I did want to tell you about another sort of sensory restoration process in vision that deals with gene therapy, but that's not through optogenetics. And in this particular case, the focus was to try to see if anything could be done to give people with only 2 cones normal color vision. Because remember a couple weeks ago, we talked about how if you have only 2 of the 3 cones, you are colorblind in a variety of ways. So could anything be done to, for example, if people up here who are red-green colorblind, give them the red color cone back? So it turns out that there is a species of spider monkey where the males are missing the gene that creates the red cones, okay? So they are red-green colorblind in the way that dogs are, in the sense that they have the cones that are associated with blue and the cones that are associated with green, but they don't have the ones associated with red. So what you can do is you can try to get the monkey to touch the little part of the screen where there's color, and it turns out the monkey isn't going to be able to do that. And so if the monkey, you know, if gets it in the right third, you know, it could be in this third, or this third. If the monkey presses the right third, it gets a little treat. In this case here, the male monkey is lost because it tried to give it in the middle because it just can't see the red because it is colorblind. So it actually turns out that what you can do is you can take the gene not from the males, but from the females, because the females do have the gene that causes or the formation of the red cone. You can put that gene in a virus, just like we did with optogenetics, and then you can literally inject that gene into the eye of the monkey, okay? And then the virus will infect the eye, it will cause the infection to create some red cones, and then maybe you can restore color vision. So let's watch a little video about that.
So humans and other old world primates have two genes on the X chromosome that encode visual pigments. One encodes the red cone pigment and the other one encodes green cone pigment.
But if you're colorblind,
only one type, red or green, is expressed.
finding that faulty gene was relatively simple. Replacing those genes inside the cone cells with the right genes, well, that's a bit trickier.
You have to have some way of delivering a gene to the cells that you're trying to treat and not into other cells.
Fortunately, nature has crafted a really powerful method of forcing DNA into a very specific cell, a virus.
The virus that we use is called adeno-associated virus, and people call it AAV. Its main advantage is that you don't get an immune response against the virus.
So while researching how to load the therapeutic gene into the virus, the Knightses trained a pair of colorblind male squirrel monkeys, Sam and Dalton, to take a colorblindness test.
Every single morning, the monkeys wake up, and before they have breakfast, they go, okay, it's time to have our color vision tested. And the monkey is trained to touch the place where they see, that colored blob, and then they get a treat. They can be most efficient and get the most rewards if they're, just touch with their nose and then they get down and, get their little treat.
And just to be thorough,
we also ran untreated animals. Occasionally they might just by chance touch the right spot, but over trials, that they can only really get it right all the time if they have normal color vision.
Once the monkeys were trained and the virus was ready, Sam and Dalton underwent a fairly elaborate procedure.
A vitreoretinal surgeon slipped the needle underneath the retina. Then the fluid is infused in order to treat the whole entire back of the eye.
It wasn't immediately obvious that it worked.
And we didn't know how long it was going to take for them to change their behavior after the pigment was expressed robustly.
Now when you look back and you see the difference between the animals, it's so dramatic, It's an amazing thing, and it amazed us.
Although the FDA has yet to approve the procedure for human trials, recently the Knightses have developed a one-shot version of the cure.
It's like an everyday shot that would take, 1 second, just a shot right into the eye.
And while getting a shot in your eye sounds terrifying to some, it's a small price to pay for living out a dream, or getting to see a sunset in all its glory, or maybe just not leaving your house dressed like this.
Yeah, so actually, the man in the video, that's a couple. They're I think the name are what is his wife's name? Sheila? I know Jay, but I don't know his wife. But Jay Knights. I was talking to him once at a conference about this. I was telling him that I often teach it, and he was telling me about how there are people who sometimes contact the Knightses and want to volunteer to be the first human guinea pigs, and they're like, "Well, no, sorry, it's not FDA approved. We can't do that." And people have literally been like, "Well, if you happened to have left the vial on the counter by the front desk and you happen to have forgotten to lock the door on your way home, it wouldn't be your fault that someone came in the middle of the night and took this and injected it in their eye, right? Wink, wink, nudge, nudge, eyebrows, eyebrows." So people are actually really excited by this, and people who are colorblind really love the idea of being able to get this treated. I love the idea not only of just being able to treat it to restore a sensory experience, like, give you red and green perception if you're colorblind, but actually potentially expanding the senses. What if you took the gene from a butterfly, which has, ultraviolet perception, and gave it to us because we see new colors that humans have never seen before? All right, so that's just a brief foray into sensory restoration. Like I said, we'll talk about it a little bit later when we in the domain of audition, but we'll save that because to understand how cochlear implants work, you really have to understand the auditory system, which we haven't gone into yet. But since we have a good understanding of the visual and motor and somatosensory system, I thought we could talk about this here. That is all for now. Actually, next week, we will talk about audition and cochlear implants. We're going to talk about sound, smell, and taste. We're going to talk about all three of these actually in one given week. The reason we're going to do all three in 1 week is actually they're relatively poorly understood and much less studied than movement, touch, and vision. So we'll stop there. I'll see you all in discussion section pretty soon. All right, take it easy.